talk a little bit more about glycoproteins. They tend to have very heterogeneous glycosylation. You can analyze them by UPLC methods. So here we've sort of analyzed all the glycans from fettuin, and you can see they have all these different sequences. But we wanted to ask the question here whether an enzyme that selectively cleaves one particular sugar off was actually selective. And what we wanted to know is whether this is silyl lactose, whether this enzyme would cleave one of the two isomers selectively over the other. So one very specific question. So we took this whole mixture of glycans, fragmented it by our fragmentation method, and then looked actually for the ion that represented this particular target structure, which we were really interested in. This is actually a terminal structure in many of these glycans. And we could see that when we treated it with our enzyme, actually we started with fettuin here, sorry, this is not on order, with fettuin, which has both linkages. When we treated it with, we had a simple glycan, we could show that this here corresponds to one of the anomers. Our enzyme cleaved the other one off, and the third enzyme here cleaved mainly this structure off. So really, you can get sequence information, very selective sequence information from very, very complex samples. The last example I want to show is about plant glycosides. These are particularly difficult to sequence because we don't have any of these nice LC methods we have for N-glycans. And here we worked with some plant biologists, Birger Möller at Carlsberg Laboratories, who was interested in plant glycosides that had these alpha-hydroxynitrile A glycones. So these structures, so this is, hold on, this just tells me that I've done 20 minutes, so 5 more minutes and we finished. So basically, once you cleave the sugar off, this generates cyanide, and that acts as a defense mechanism in many plants, for example, in almonds. So they were very interested to get the structure of these glycosides. And some of them they had determined, and you can see they're very complex. And I think this is not uncommon. I'm not a plant biologist or biochemist, but I think this is not uncommon that you have plant metabolites. Notice there are also different glycones here. These nitriles come in different forms, but you also can see there's quite a lot of heterogeneity on the sugars. And what they really want to know is what is the sugar composition of these and what are the linkage types, and that's a very, very difficult answer to give. So what they normally do is they extract the plants, they do LCMS, then they do prep HPLC of the major peaks. The minor peaks they don't get enough material on, and then they have to do either NMR or they have to do chemical synthesis from authentic standards and compare them with LC methods. So this is a very, very time-consuming method. So the question was, could we use our fragmentation method now to really do de novo sequencing of these plant metabolites? And to finish off one paper, they had this one challenge, that they had these glycosides of this natural product here, amygdalin, from almonds, and the LC gave them 3 peaks, and from the mass spectrometer, which from LCMS, they could show that there was a hexose linked to this structure here, but they didn't know which hexose, and they didn't know what the regiochemistry was, and they didn't know what the linkages were. But they realized that these 3 were actually isomers, 3 isomers of this general structure. So they challenged us to use the sequencing method to actually find that information. So we did the same thing. We started with our target structures. We could get them straight from their LC. And then we fragmented them in our mass spectrometer and did ion mobility separation. And what we obviously had already were standards that corresponded to substructures of this target structure. So we didn't have any authentic material of the target structure, but we had substructure. We had this amygdalin here, and we also had these diglucosides, which I've shown before here, which you potentially could correspond to these terminal structures. And by comparing fragmentation and ion mobility spectra of the target molecule with these standards, we could unambiguously show, and these are sort of the data, which I don't want to go through. In fact, this was published in Biochemical Journal. We could actually tell them, that this here, they all three have glucoses attached to them, because it clearly showed that the ion mobility was corresponding to glucose. They were all beta linkages. Again, we can have this anomeric effect that we have the memory effect, and that this one here is linked to the 6th position, this is linked to the 4th position, and this is to the 2th position. And really, at the moment, we don't have any other analytical methods which could do this, at this sort of concentration from crude mixtures. So this is a really nice way of showing that in principle these methods are really giving us useful information. I should also say this also works with N-glycans, so we've taken this typical core pentasaccharide structure. With N-glycans, as I said before, we have other analytical methods, but it's a generic method that also works for N-glycans, and we can fragment that into these, and then we can compare that again to standards, structures, and all these collisional cross-section values correspond very, very nicely to those of standards, generated from completely different starting materials, so it's obviously independent of starting material, and then you can assign actually the structure by building up from the fragments. Obviously that takes quite a lot of effort. So just to conclude our sort of vision for developing carbohydrate sequencing method, it's not that we maybe come up with a very, very simple method, but what we find is that incorporating ion mobility with already existing mass spectrometry methods means that sequencing, hopefully, will become much more generic. So we're at the moment, we get from mass spectra what we call sort of two-dimensional information. We get already quite a lot of information on linkages, whether you have hexoses or hexnags, what the molecular weight of the building blocks is, whether it's branched or linear. We can do that already with existing mass spectrometry methods. But what we can't do is get the three-dimensional structure, and at the moment we have to go through these protocols, and we need very, very large databases. There's currently a program funded by NIH which asks chemists to synthesize all the potential glycan structure in the human glycome. So these are 10,000 different structures. So we really need that if we pursue these sort of LC-driven processes. If we incorporate ion mobility, we propose that we go to the fragment level. We don't need the LC because ion mobility acts a little bit as an LC, but the beauty about ion mobility is we can do it on these fragment ions. We need much smaller databases, and hopefully that will really open up the area and allow us to develop much more generic sugar sequencing protocols, so that when people isolate carbohydrates, they can get very easy, very high resolution and accurate sequence information. I should say at the moment, you can see we're dealing with very simple structures, so I always compare the state of this sort of sequencing to the first commercially available TV, which is this one here, which was commercialized by the Baird company at Hastings in the UK in 1930. So this is what the first TV looked like. So I think we're still quite a way to match actually sequencing of DNA and proteins, but hopefully we're getting there by using this methodology. So I just have to thank really my team, in particular for the sequencing programs, Chris Gray, who was an absolutely outstanding PhD student, my collaborators who are mass spectrometrists, Claire Ayers and Perdita Baron, in particular Birger Moller for that collaboration on the plant project, funders, and I thank you for your attention.
Thank you, Sabine, for a beautiful talk, inducing us to do these simplified methods in the future.
Thank you.
Any questions? Please.
Very nice talk and very informative information related to the stereochemistry, because from the long time that carbohydrate chemistry are trying to solve this the problem related to the stereochemistry, variation of the stereochemistry of this glycan. Just two things I just want to know, that is it this technique, this ion mobility technique is still will be applicable for the biopolymers like xylan or starch, so these are the bigger the biopolymers, or it is only for the oligomers? And the second is, if it is for oligomer, then what the extent of the DP, the degree of polymerization that to which we can analyze the oligomers. So that is the thing.
Yeah, that's a very good question. I mean, I think the polysaccharides will be a big target. I think we probably have to do pre-fragmentation of these, maybe through chemical methods, and then fragment at the fragment level. I mean, I think the DP, given that we deal with mass spectrometry here, I haven't shown a lot of other. But you can actually do the ion mobility on much larger fragments, as long as you have the standards. So you're absolutely right. The big challenge is in the polysaccharide field, and that's where we're moving. And this is why I showed this primitive TV. I'm not trying to sell this as at the moment state, ready to go. But polymer, we're working at the moment on polysaccharides. I think that's a really, really good challenge area. I agree with you.
Because either they have to be some chemically defragmented or something prior to probably to this kind of analysis.
Yeah, or enzymatic. It depends how big they are. Yeah, we can already do some fragmentations, but we haven't really done very much in that area. So that's sort of the next step. We focused on smaller structures at the moment, but it's sort of bottom up. But it's definitely something we're thinking about. Yeah, I think it's a very important application. Yeah.
Okay.
Let's give another hand to Sabine Flitsch. Thank you, Sabine. The next speaker is Nico Kallevart, and he also was introduced as former chair already, and he will partly continue the topic that was discussed currently.
Well, thank you so much. It's a real honor to speak here for this fine audience, and I have to thank really the organizers locally for doing a splendid job. I'm representing a few people who do not travel much anymore, and so at Ghent University, where I work at the moment, I'm the third generation of people who have had some contact and training by Gobind. And so the other two people would be Walter Fiers, who was with Gobind in 1962 already. And he was the PhD supervisor of Roland Contreras, who was with Gobind in 75, 76. And Roland was my PhD supervisor. And I was sent to MIT for a brief while in 2000 or 99 it was, to try and help out with a particular challenge that the lab was facing at that time. So Walter really, to just illustrate the impact that Gobind has had also in our country. Belgium is a very small country, and the northern part is Dutch speaking. It's called Flanders, and we have a biotech institute there since about 20 years. And Walter was one of the founding fathers really of that institute. And there's now two centers, the Inflammation Research Center and the Medical Biotechnology Center, which I head at the moment, that really come directly from research lines that Walter has started up. And the Inflammation Center is built on the early work on cytokine gene sequencing that Walter did. And then the Medical Biotech Center is very much protein focused and derives from the sequencing work and the heterologous protein expression research line that Walter started very early on. So we're very, very grateful for that legacy in our country, really. So what I want to bring scientifically to you today is two stories that deal with, I think, big problems that act beyond the ribosomal stage of protein synthesis in eukaryotes. And so that would be processes that occur in the eukaryotic secretory system. And very broadly, conceptually, what happens in the eukaryotic secretory system is that after a protein has been translocated into the ER lumen through the translocon upon ribosomal translation, then glycans are attached, depicted here with these monosaccharide symbology that you just saw in Sabine's talk as well. So oligosaccharides are attached in glycans to the asparagine side chains of particular sequence in the protein. And these glycans play essential roles. They're universally conserved in the eukaryotic world. They play essential roles in catalyzing protein folding in the ER lumen. And they do that through association of particular intermediates, processing intermediates of the carbohydrate with chaperones, such as calnexin and calreticulin. And then after the protein has attained its proper fold, then the protein is exported from the endoplasmic reticulum into the Golgi apparatus, where then a species-specific array of glycosyl transferases is present that takes that universal mannose 8 GLUCNAC2 precursor and remodels it into species or even sometimes cell-type specific glycosylation structures. So there's a whole array of those, and then the final product would be a glycoprotein, and here represented by the influenza virus hemagglutinin, which is the main antigen in the current influenza vaccines. It's here in complex with a Fab fragment to help it crystallize. But I like this picture because it illustrates that carbohydrates, everything in yellow here is carbohydrate, are really very large modifications that cover up extensive areas of the protein surface. And so if you want to use a protein like this biopharmaceutically as a vaccine, the naive B-cell receptors have almost no chance of recognizing epitopes that are partially shielded by these carbohydrates. And as a matter of fact, quite a few viruses use this as an effective evolutionary strategy to escape neutralizing antibody responses. And the HIV virus is a case in point, but the influenza virus does that as well. So that's why from this biopharmaceutical research line that started, that Walter Fier started, initially this was all in E. coli, but a lot of proteins will not fold in E. coli precisely because they lack this glycan-associated protein folding catalysis. And hence, in biopharmaceutical industry, we have to use eukaryotic systems to cope with that problem. So intact monoclonal antibodies, for example, which is 60% of the pharma pipeline today, fall in that category. And so we're not using E. coli to make those, we're using mammalian cells. So this is also the context a little bit in which Gobind called upon Roland's expertise, and he knew Roland was working in the group in Ghent on glycosylation engineering, and at a certain point Gobind was trying to express, in fact successfully expressed, rhodopsin recombinantly in HEC293 cells, human embryonic kidney cells. But then a major problem that people were confronted with in using those cells for structural biology was that you get tremendously heterogeneous glycosylation on these proteins. And this is the protein as you would extract it from the rod outer segments from bovine eyes. But the protein you make recombinantly is really tremendously glycosylated. And here is the glycan. It's difficult to see. Really, the way to look at this would be that the sugars will be as big as the protein if they are made in HEC cells. So that was a problem, and then Phil Reeves was working on that, and I just got in touch with him yesterday. He sends his best regards. The reason he couldn't come is that he's really very heavily occupied in teaching, and his university wouldn't let him escape, even for a few days now. So the way Phil and I started working on this is based on a trick. And if you mutagenize mammalian cells, there is a slight chance that you might hit all of the alleles in HEC cells that code for this particular glycosyl transferase called GLUCNAC transferase 1, which is really the gatekeeper in converting high mannose carbohydrates into so-called complex type glycans at the cell surface of mammalian cells. And then ricin toxin has a galactose binding domain, and it has of course a toxin domain as well. But ricin toxin cannot get into mammalian cells if there is no terminal galactosides at the cell surface. So if you hit all of the alleles here of GLUCNAC transferase 1, you generate a cell that can survive ricin selection. And so that's exactly the way this was done. And then this results into a protein that is really very homogeneous indeed, pretty much similar to what you would extract from rod outer segments. And then this is a carbohydrate analytical method that I developed during my PhD, and perhaps that was appealing to Gobind because what we used was a sequencing device from applied biosystems that had been developed for Sanger sequencing of DNA. So we kind of we labeled these carbohydrates with anionic fluorophores to make them move into the capillary electrophoresis, so in the electric field. And then we used that DNA sequencer with laser-induced fluorescence to map out these carbohydrates linked to the protein. And so what you get here is a size standard, which is a hydrolysate of starch. And then this is the glycans that you get from the wild-type HEK cells. As you can see, this is very, very heterogeneous. And then from rod outer segments, you get this profile. And then from this glutamate transferase 1 knockout mammalian cell, we got a very homogeneous profile indeed. And so this generated a paper that I checked yesterday. It has been cited 450 times by now. A lot of structural biologists are using this cell line to cope with similar problems that they face. Okay. So then I want to switch gears a bit, and the rest of the talk will be about yeast. And so yeast has been an organism that we've worked on for the past close to 20 years now to see if we could actually modify the glycosylation pathways in this organism so that they would correspond to what mammalian cells normally make. And the idea here is that at present, the biopharma industry is using mammalian cells, which are really not very robust cells, if you look at them carefully. They don't have a cell wall. They need expensive media to grow. They require extensive control of their extracellular environment to cultivate them. And so if we could manufacture glycoprotein therapeutics in yeast, that may be a reasonable advance and would reduce costs and allow us to do the synthetic biology approaches that we've heard about in a microorganism now rather than in a mammalian cell to try and optimize pharmaceutical protein production. And so we've been looking mainly at the types of glycans as you find them on the CH2 domain of human IgG1. So there is a conserved glycan there in glycan of fairly simple structure. So they're biantennary N glycans that carry a core fucose residue, which is important for the interaction with Fc gamma receptors. And then there's partial galactosylation of those branches. So we tried and built yeast to an organism that could synthesize something like this. And so yeast obviously have a cell wall, which makes them very robust as expression systems, but at the same time causes a bit of a challenge for us because the cell wall is largely made up exactly out of those glycans that we now try to engineer. And so you have to walk a narrow path between modifying them to the human system and yet make sure that the yeast is not suffering from that too much. That's why perhaps it has taken us more than the 2 or 3 years that initially we thought it would take. It's taken us close to 2 decades to solve this to our satisfaction. So the pathway really in the Golgi differs between mammalian cells and yeast cells. What yeasts do is to take this universal mannose-8 glutamate-2 product that comes from the endoplasmic reticulum, and then it extends this with further mannose residues and mannose phosphoryl residues, and really build extensive structures that form part of the cell wall. Now, what we try to do is to knock out this initial transferase here, and then we introduced a fungal mannosidase to convert to mannose 5. And then we built in the different glycosyl transferases fused, so the catalytic domains from mammalian glycosyl transferases, but fused to the Golgi targeting signals of fungal glycosyl transferases. And in exactly how you make that fusion, and in exactly how you tune the expression levels of those enzymes, that is where the difference is between a proof of concept experiment and something in which this pathway is 90 to 95% efficient at every single step. And that took us a long time to accomplish. So in around 2008, 2009, we thought we were more or less done. And here again you see capillary electrophoresis profiles where this is the wild-type glycosylation, MAN5, glutamate MAN5, and then glutamate MAN3, glutamate2 MAN3, and then the galactosylated form. And in fact, we thought this looks like the glycosylation profile that you normally find from CHO cells that make monoclonal antibodies. And Merck Pharmaceuticals bought a company called Glycofy as well that we were competing with at that time. And Merck paid close to $500 million for this at this stage. And we were so surprised because around that time, we discovered that the job was not quite done after doing this. And the point is that if you start looking more carefully at this, what first looks like a clean baseline, there is actually tiny vibrations of that baseline, which indicates still remaining endogenous yeast glycans. And if you add them all up, you end up with 25 to 30% of the total glycan profile. And so what Merck scientists tried to do was to knock out one after the other yeast glycosyl transferase to try and get rid of that background. But in the end, you have a very sickly yeast indeed, and so that didn't get them very far. Even more worrisome, what you sometimes see on particular glycosylation sites, on particular proteins, is that all of a sudden, the human intermediates that the yeast evolutionary has never seen before start becoming substrates for glycosyl transferases that still sit in the Golgi of yeast. Pretty much, I could relate to what Dr. Zo was saying. These enzymes have never been selected before in evolution to not recognize things that they have never seen before. And so it took us about 2 years to figure out what this was. This is a structure that has never been described before. We're just publishing it. And if we had had the methods of Sabine Flitsch, as you just have heard about, this would have sped up this research tremendously. And so this is very important indeed. So now, after quite a few years, and then, from Gobin I heard a lot here, and I've been taught by Walter and Roland as well, never ever give up, right? And so one day there will be a new idea that may solve these kind of problems. And indeed, such a new idea came around when we learned that certain fungi actually secrete their glycoproteins, not with this kind of sugars, but with a single GLUCNAC residue on them. And that made us think, oh, they must have an enzyme that can take that off. And that enzyme is expressed in a fungal organism, so clearly it's not toxic to a fungal organism. And so we cloned it, and then with another 3-4 years of work to finally tune the expression of that enzyme, we could manage to express it in Pichia pastoris, which is the yeast that we are using here, metallotrophic yeast. And so in a single step modification, in fact, this is GM-CSF, granulocyte macrophage colony stimulating factor, an important therapeutic. It normally has, it has 2 N-glycosylation sites that are partially occupied. This is the non-glycosylated form, 1 N-glycan and 2 N-glycans. And then you see the heterogeneity typically associated to yeast and carbohydrates. So in the glycodelete, as we call this system now, with a single transformation, we can get rid of all of that. And this is fully compatible with yeast life. It's not always as simple. Sometimes we have tremendously heterogeneous products, like interleukin 22 in this case, which is also proteolyzed partially. But at least we can convert this, like probably thousands of different glycoforms into 3. So this is a proteolyzed form. This is the intact form with 3 times single GLUCNAC. And this is a form which we think is a part of the pro sequence, the secretary signal that we use there. So at least here you can purify. I mean, you can forget about trying to purify anything out of that there. So we combine that now with what we call this Glycol Switch, because it switches from yeast to human. We combine that now, and so it's a kind of like a restriction enzyme, if you wish, for carbohydrates. So everything which remains of the yeast type is now substrate for this endoglycosidase, such that these structures are all converted to a single GLUCNAC. And this enzyme that we use, this glycodelete enzyme, does not recognize the human intermediates. And so those remain on the protein. And so really this allowed us now, as you can see, you see all these tiny vibrations on the baseline and some intermediates there. If we combine this with glycodelete, all of that is gone, and these remaining structures are these intermediates that we synthesize, or human intermediates. So now, after all this work, I think I now dare say that this system is ready for antibody production in biopharmaceuticals. It was not 10 years ago. So what could we use this for now? And this is a bit of a future perspective, perhaps. I think we've all witnessed the outbreaks of Ebola in Western Africa and the outbreaks of influenza more recently. And there, the drama was that we had antibodies to protect these people. We knew which antibodies to use, but we couldn't roll it out quickly enough because there was no production methodology that would allow us to respond quickly enough to this. Now, the good thing about yeast is that it has a doubling time of 2 hours rather than the 24 hours that you find in CHO cells, allowing us really to make, if we have broadly neutralizing antibodies, which we have, to